Welcome to Biology 1440. This lab is Lab 2 in your manual. Hypothesis formulation, experimental design, and data analysis, making a standard curve. So this lab and the next lab really focus on cell membranes and how they might be damaged and how we could measure that damage. And cell membranes are composed of a phospholipid bilayer with proteins embedded within, and you'll learn more about that in a lecture. But it's something worth studying. And we're going to be using beets, garden beets, as the organism in our experiment. And why in the world would we use garden beets? Who cares about beets, right? But garden beets are a great model organism. A lot of times scientists will use a model organism to study a system because the organism's easy to manipulate or offers some benefit for the experiment. In this case, beets have a cell membrane that looks quite a bit like ours, quite a bit like those of a bacterium as well, or even the phospholipid layer that surrounds the coronavirus. So if we can measure how we can damage it in a beet, we might be able to better understand what damages cell membranes in other contexts. And beets have a red juice inside called beta-cyanin that leaks out when you damage their membranes, making their damage easy to detect, which is why it's a great model for studying cell membrane damage. Here's a picture of a plant cell from your textbook. There is an outer cell membrane called the plasma membrane, which is shown here. Don't confuse the membrane for the cell wall. Those are very different things. The cell wall, think of it as being made of wood. The plasma membrane is more like made out of cooking oil, okay? And here's the plasma membrane, and then all the organelles inside the cell are surrounded by plasma membrane as well. Obviously not plasma membrane, but by phospholipid bilayers as well. And there is a phospholipid layer around this organelle here, which is known as the central vacuole. The membrane here is not called a plasma membrane, but in this case it's known as the tonoplast. So the tonoplast is the membrane around the central vacuole. The central vacuole fills full of liquid and helps plant cells keep their turgidity. So when you have like a piece of celery that's nice and crispy, it's because the tonoplast is full of water and it's got a lot of pressure in it, and that pressure is pushing against the cell wall, giving that piece of celery a lot of rigidity. In the case of beet cells, they store beta-cyanin in here, that red stuff that leaks out. So if we damage this plasma membrane with, let's say, ethyl alcohol, maybe we dissolve the phospholipid layer with alcohol, or maybe the alcohol denatures proteins in that layer, then the alcohol could get in here and denature the proteins in the tonoplast membrane, or dissolve the phospholipids here, allowing beta-cyanin to leak out into our test tube, and then we could see that damage or measure that damage. So if we were in person this week, we would be learning to use a pipette. We would use the mass balance equation, which I do expect you to work on. You would use a spectrophotometer to record light absorbance in various solutions of beta-cyanin and create a standard curve showing the relationship of light absorbance at lambda max and beta-cyanin concentration. Now, I don't expect you to know all this terminology yet, but we'll get there, okay? So first of all, you're going to be making a standard curve. What is a standard curve? It's a graph. It's much like the graphs you've made already in this lab. We'll use the scatter plot feature in Excel to make it. It's a graph that you make, first of all, using standards. Standards are, in our case, beta-cyanin concentrations of known concentration. So you'll make a graph of how much light they absorb. And so one axis will be what their concentration was. The other axis is how much light they absorb. Those are standards. You know what their concentrations are. The important thing is then, if you're given an unknown concentration later, you can use your standard curve to figure out the concentration of the unknown through interpolation or extrapolation. So if this is an example of, here's our data points, let's say we make a graph, we put a best fit line on it, this is our standard curve. And the units have no real meaning on this example. But let's say we give you an unknown concentration, okay? So some unknown concentration of beta-cyanin, and it falls, right here on the graph. Well, you'll be able to figure out what that concentration is using your standard curve. So for example, if you know that the y-axis is 60 for the unknown concentration, you can draw a line straight over to the best fit line and straight down, and you'll know what the x-axis should be for that unknown. Or even more accurately, if you know y, you can solve for x in the equation of the line. Y equals mx plus b. You can solve it that way. Now, if your unknown falls within the range of your standards that you use to make the graph, then solving for that unknown is known as interpolation, and it's probably pretty accurate if you do a good job. If that unknown is way more concentrated or way less concentrated and falls outside of the range of the standards that you use to make the graph, then it's called extrapolation when you predict the concentration of the unknown solution. So here are some images of some beet cells. You can see the beta-cyanin inside of these things, the red pigment. And what we would do in this lab is we would grind up these beets and just give you some straight beta-cyanin solution, so beet juice basically, and you would run that juice through a spectrophotometer. Spectrophotometer is a device that uses visible light to measure concentration. So visible light falls within part of the electromagnetic spectrum here. It's a very narrow part of the electromagnetic spectrum, and it ranges from 750 to 400 nanometers in wavelength. Wavelength is the measure from one wave to the next. Down here, low energy red light is at 750 nanometers. That's how far it is between the wavelengths. And way up here, 400 nanometer violet light. And note that the symbol here for wavelength is this upside down Y looking structure. This is known as lambda. This is a Greek letter lambda. So lambda is the symbol for wavelength, okay? And the wavelength that is most absorbed by a substance is known as lambda max, and that wavelength varies from substance to substance. So because different substances absorb light at different wavelengths, we can use a device known as a spectrophotometer to detect the substance and to calculate its concentration. So a spectrophotometer is a device that has a light source and then perhaps a prism or different color light bulbs. So it shines light of different wavelengths through the sample and then detects which wavelengths pass through and which wavelengths are absorbed. And just so you know, like in this example, the beta-cyanin would be in this cuvette right here. A cuvette is the fancy word for the container that holds the sample in a spectrophotometer. So here's an example of what it looks like when you put beta-cyanin of different concentrations in a spectrophotometer. So these were recorded one at a time, but I saved all of the different samples on the same graph so you could see. So notice we have wavelength down here from nothing to 700 nanometers. Here is absorbance. That means how much light was absorbed, and absorbance has no units, okay? And you can see right here is where light is most absorbed by beta-cyanin, right around 535 nanometers. And notice what happens when you dilute beta-cyanin. So this was the most concentrated solution. Down here is the least concentrated solution. Notice that the lambda max, the wavelength most absorbed, does not change. It does not move left or right, is what I'm saying. It doesn't become more blue or more red. It stays at the same the same color, I should say, the same wavelength. But the amount of light absorbed changes. As you dilute the substance, the amount of light decreases, but it doesn't shift left or right. That's a really important fact. That's why you can use a spectrophotometer to determine what the substance is and what its concentration is. So you might use a spectrophotometer, let's say, in a crime lab to determine what poison killed somebody, or you might use it in chemistry. You'll use it in several courses probably throughout your academic career. Another tool you need to understand this week's lab is the mass balance equation. This is an equation that is used to figure out how to make dilutions. Okay, and it works basically on the basic premise that C1, that's the concentration of the stock solution, that's the solution in the back of the lab, let's say, or the solution in a container, times the volume of the stock solution will equal the concentration that you're making times its volume. All right, so the concentration of the stock solution plus how much volume you needed to make your dilution, which I'll explain more in a minute, equals C2 times V2. So basically you'll hear me say C1 times V1 equals C2 times V2. I just want you to see what those words mean here. And you can see it kind of written out down here. Now this is important because it lets us make dilutions, and we can do it quite accurately using our pipettes and this equation. So for example, how much stock solution did you use of 80% beta-cyanine when you make 20 milliliters of 20% beta-cyanine? All right, so how in the world would you solve this? Well, first of all, you figure out what your variables are. I don't know if my pen will work here. It probably will not. No, it's not working. Okay, so how much stock solution did you use of 80% beta-cyanine? 80% beta-cyanine is what you used. That's your stock solution, okay? That's the solution in the back of the room. When you make 20 mils of 20% beta-cyanine. So 20 mils of 20% beta-cyanine, that's what you're making. You're making 20 mils of 20%. So this is V2 and this is C2, concentration to volume to. Remember, volume is how much, concentration is how concentrated it is. So you know the variables now. This is C1, this is V2, this is C2. So what we can do then is, if we remember that this is basically just basic algebra, V1 equals C2 times V2 over C1. Because remember, the basic equation is C1 times V1 equals C2 times V2. So we figure out which one we know and which one we don't know, and we can set up a very basic equation to solve for the unknown. So in this case, the answer is 5 milliliters. So we take, in this example, 20 times 20 over 80, okay? And then we know we need 5 milliliters of stock solution to make our dilution. All right, here's a slightly more complex one, but a very important one. You'll have questions like this on most of your lab quizzes. You use 90 molar ethanol to make 15 milliliters of 10 molar ethanol. How much did you use, and how much distilled water will you need? Okay, so once again, assign your variables. You used 90 molar ethanol. Molar is a concentration, so this is C1. To make 15 mils, okay, you made it. That's a volume, so that's V2, and this is V1. How much did you use? Okay, set it up. So once again, you'll take this times this over this, right? It's the same equation as the last example. C2 times V2 over C1. So you get 1.67 mils. Okay? All right. So we know we made 15 mils, and we know we used this much stock solution. Notice these two numbers are very different. We used this much of this stock solution to make this, right? So to figure out how much water we added to this, we just simply find the difference between V1, volume 1, and volume 2 here. What's the difference between the two? You got to do a little math, a little subtraction, and we get 13.33 milliliters of distilled water. VDH2O stands for volume of distilled water. That's how much water was needed to make our dilution. The only way to get comfortable with this is to practice it. And you just need to sit down. You can make up your own values. There are examples in the lab manual. Sit down and do it. It's not a hard equation, but students sometimes freak out when they see math and they refuse to learn it. You have to sit down and just do it 10, 15 times until you're comfortable with it. Okay? So that's part of your homework, is to do that. There are more problems here. I'm going to skip over them for the video. Okay. Finding lambda max. So the stock solution in our example that you'll be using for homework is 16 micromolar. Okay, 16 micromolar solution. What you would do is you would place 3 milliliters of that into a cuvette, and then you would also put 3 mils of distilled water into a separate cuvette and blank the spectrophotometer. That tells the spectrophotometer to ignore water but only measure beta-cyanine. Okay, and then you would put the cuvette in the spectrophotometer and you would record hit record, basically, hit collect data, and you would get a peak just like we saw in the previous slide, and you would see how much light is absorbed at that peak wavelength. And I'll have a video on that here in just a little bit. So here's an example of a dilution table that we would make if we were meeting in person. This is the mass balance equation in table form. So C1, V1, C2, V2, and volume of water needed to make the dilution. So what you would do is you would take your stock solution of beta-cyanin, which is 16 micromolar, and you would dilute it to make different concentrations, your standards, your known concentrations. You would make those, and you would put each one in the spectrophotometer to see how much light each one absorbs. And I started this table for you, but I left a lot of it blank so you can practice the mass balance equation. So, for example, our most concentrated stock solution, C2, is 16 micromolar, which is the same as the stock solution. So we would make 16 millimolar. It's the same as the stock. So I did that one for you so you can see how the math works there. Our lowest concentration is 0 millimolar. I'm sorry, I keep saying milli. 0 micromolar beta-cyanin, which is basically pure water, right? And I did this one as well so you could see how the math works. So C1, V1, C2, V2. For the rest of these, this is the concentration you would have to make, and this is the stock solution. So you can work on this to see if you can figure out the logic of the mass balance equation to fill in the rest of that table. So we're making, just to be clear, this is the stock solution concentration. This column is how much stock solution you need to make the different dilutions. This is the concentration of what you're making, C2. This is the volume of how much you're making. And this column is how much water you would need to add to this concentration that you're making, or to add to the stock solution, I should say, to get the concentration that you desire. So ultimately, you're going to then make a figure of this data. So graph, caption, best fit line. And there's an assignment online that I want you to work on. You'll download the sheet and you'll answer the questions on the sheet. And be prepared for a quiz when you come to lab in the future. Okay? So your figure is going to look something like this. It's going to have beta-cyanin concentration on the x-axis and absorbance on the y-axis. Okay? So that's basically what your graph is going to look like, and it'll have your different data points and a best fit line on it. I want you to also insert the equation of the line onto that graph. It's very important that you do that. And there'll be a few questions then on your worksheet to answer after you've made your graph. All right, so I'm going to have a little video clip on, once again, how we collect the data in lab to give you a better indication of how that works, and then we'll call it a day and you can get started on your lab. So here we see the spectrophotometer, a pipette, the beta-cyanin solution, and a cuvette. We take the cuvette and we make sure that the rough edges are facing away from the arrow on the spec. We place the cuvette into the spectrophotometer and we click the collect data button on the program. So now it's going to record which wavelengths are most absorbed, and there we can see the data. We can see the most absorbed wavelength there is in the green spectrum, around 535 nanometers. I then go to the examine tool and I find 535 nanometers using that tool, and it will tell me exactly what absorbance is at that wavelength on the screen. And that's how we would get the data that you'll see on the worksheet.